Welcome back to the Quantum Leap. I'm your host, Dr. Nirdosh Jagota. In our previous episodes, we've talked extensively about the software side of the AI revolution in pharma. We've discussed algorithms predicting protein structures, neural networks generating novel molecules, and machine learning models mapping cellular biology. But drug discovery isn't just a software problem. At the end of the day, a drug is a physical molecule. It has to be synthesized in a lab, purified, and tested against physical biological targets. And historically, that physical process, the actual chemistry, has been the ultimate bottleneck. Traditionally, if you want to discover a single drug suitable for clinical development, a team of human chemists might need to synthesize between 2,500 and 5,000 different molecules. That process alone often takes 5 years or more. It is slow, it is incredibly expensive, and frankly, it is prone to human bias. But what if we could automate the chemistry? What if we could build a laboratory that drives itself? Today, we are exploring the rise of the self-driving lab. And to do that, we're going to focus on a pioneer in this space, Exscientia. The story of Exscientia begins with its founder, Andrew Hopkins. Back in 1996, Hopkins was a PhD student working late into the night in a lab, trying to find drugs to treat HIV. HIV is a notoriously adaptable virus, often developing resistance to new drugs within weeks. During one of those late-night sessions, Hopkins had an epiphany. He realized that if the process of drug discovery could be automated using artificial intelligence, the rate at which scientists found new treatments could accelerate rapidly enough to outpace the disease. He spent a decade at Pfizer, then held chairs in medicinal informatics and translational biology at the University of Dundee. Finally, in 2012, he spun out Exscientia with a clear, audacious mission: to automate drug discovery in its entirety. Hopkins has famously referred to this vision as autonomous driving for the pharma industry. And Exscientia proved the concept worked. In early 2020, they announced that a drug they created for Sumitomo Pharma to treat obsessive-compulsive disorder had entered clinical trials. It was the first time in history that an AI-designed drug candidate had entered human testing. And crucially, it took them just 12 months to discover it, a fraction of the industry average of 4.5 years. Since then, they brought several more AI-designed drugs into the clinic, spanning oncology and immunology, and they've cut the number of molecules they need to synthesize down to just 150 to 250 per project. But how exactly are they doing it? The answer lies in closing the loop. To understand the self-driving lab, we have to look at the core engine of drug discovery, the design, make, test, learn cycle. First, you design a molecule. Then you make it, you synthesize it in the lab. Next, you test it against your biological target. Finally, you learn from the results to design a better molecule for the next round. In a traditional lab, this cycle is disjointed. An AI might design the molecule, but then a human chemist has to figure out the synthetic route, manually mix the reagents, run the reaction overnight, purify the product the next day, and hand it off to a biologist for testing. It takes weeks or months just to complete one cycle. Exscientia realized that to truly harness the speed of AI, they had to automate the physical execution. They built a massive 26,000 square foot robotic laboratory at Milton Park near Oxford in the UK. This isn't just basic automation. Traditional lab automation has usually focused on parallel synthesis, making lots of very similar molecules at once. But Exscientia's approach is different. They focus on bespoke workflows that allow for a wide, diverse range of chemical structures. Here is how their self-driving lab operates. The AI model, which they call their Centaur AI platform, generates a target molecule and several possible synthetic routes. Human automation chemists review these and set up the dosing sequences. From there, the robotics take over. Automated systems run the chemical reactions. They use advanced analytical tools like ultra-fast preparative liquid chromatography and mass spectrometry to automatically monitor the reaction, extract the solid phase, and purify the final product. They are working at the gram scale to yield just a few milligrams of pure product for testing. The testing is also automated, and the results are immediately fed back into the AI model. The AI learns from the physical data, updates its parameters, and designs the next batch of molecules. They call this model-driven adaptive design. By running 10 to 15 of these rapid, automated design cycles, they can evolve a compound set to perfectly answer a project's hypothesis. A cycle that traditionally took months is compressed into days. What makes this so powerful is the elimination of human bias. Dr. Paul Cox, the director of chemistry automation at Exscientia, has pointed out that human medicinal chemists naturally lean toward specific types of molecules based on their own past experiences. They might synthesize many variants of a familiar structure, searching for the best option. But in doing so, they might completely overlook a vastly superior, entirely different chemical structure. And AI doesn't have that bias. If you encode all the available information on biological targets and molecular structures, the AI can process it and propose a small set of highly diverse molecules that test the widest possible range of hypotheses. But there is a catch, and it's a big one. A self-driving lab is not just a room full of robots with an AI bolted on top. It is fundamentally a software engineering challenge. Every instrument in the lab, the liquid handlers, the mass spectrometers, the incubators, needs to communicate seamlessly. You need a digital twin of the laboratory to simulate the physical constraints before the robots even move. You need an orchestration layer to manage the scheduling and error recovery. As we look across the industry landscape in 2026, we see this software middleware becoming the true differentiator. We are seeing the rise of vendor-agnostic platforms like Automata Link and the ChemSpeed/SciVi partnership, which allow labs to connect instruments from different manufacturers into coordinated automated workflows. We are seeing the adoption of new communication protocols like the Model Context Protocol, which allows AI agents to directly interface with laboratory hardware. The hardware is no longer the bottleneck. The robots are capable. The challenge is building the intelligent software systems that govern them. It's important to clarify what we mean by autonomous. In the automotive world, the Society of Automotive Engineers defines self-driving cars on a scale from level 0 to level 5. The pharmaceutical industry has adopted a similar scale for lab automation. Level 1 is standard automation of repetitive tasks. Level 2 involves digital protocols and machine-interpretable data. Most labs today that claim to be self-driving are actually operating at level 2 or 3. They run closed-loop optimization cycles, but a human still sets the goals and resolves the anomalies. Level 5, a fully autonomous lab operating without any human intervention, does not exist yet. And in a highly regulated industry like pharmaceuticals, it may not exist for a very long time. In early 2026, the FDA and the EMA published guiding principles for AI in drug development. A core tenet is that AI outputs are recommendations, not decisions. In good X practice or GXP environments, a human must approve any AI-driven action that affects product quality or safety. The goal of the self-driving lab is not to replace the human scientist. As Paul Cox from Exscientia notes, the ultimate aim is to create a pipeline that is human-led in terms of strategy and target setting, but operates without human interference from concept to reaction execution. The robots do the pipetting, the AI analyzes the multi-dimensional data, the human scientist directs the strategy. In late 2024, Exscientia merged with Recursion Pharmaceuticals in a landmark deal. It was the perfect marriage of capabilities. Recursion brought massive biological mapping and cellular imaging, while Exscientia brought precision chemistry and the self-driving lab. Together, they represent the future we are heading toward: an end-to-end AI-driven, robotically executed pipeline that maps the biology, designs the molecule, synthesizes the drug, and tests it, all in a continuous, rapid loop. We are no longer just theorizing about AI in pharma. We are building the factories that will manufacture the future of medicine. Thank you for joining me on the Quantum Leap. I'm Dr. Nirdosh Jagota. In our next episode, we'll step out of the lab and into the clinic as we explore how AI is transforming the design and execution of human clinical trials. Until then, keep looking forward.